Ooh. Let's try that again. It's allergy season. Hello, everybody. Good to see you. Thank you for joining us. As always, please say hi in the chat. If you haven't joined us for one of these, I am capable of forming sentences. I'm Phillip, and please say hi and tell us where you're coming from. Where are you joining us from? This is going to be a large stream today. So we have seen dynamic programming, and we've worked with it largely in the context thus far of sequence alignment, although we're not done with that. And we're going to see what we've done with sequence alignment is super useful, but we're going to extend dynamic programming to like a completely different context today, which is RNA secondary structures. And it's a classic topic in biology, showing how something that doesn't seem like it would be connected to something like sequence alignment or this concept of dynamic programming, and yet nevertheless, we're going to see that there's a very clear connection between dynamic programming and this problem. We're also going to come back to something that we've seen quite a bit, which is counting things can be fun. And you can state something that seems like it might be impossible to count. What we will see is when we get to our final problem today, we're going to have a problem that I think if you wrote it out and tried to present it to someone, they'd probably say, honestly, I don't think this will have a solution, right? Like, there's going to be so many of these things. It's not going to be clear that you could even use code to answer such a question. And the question itself is very much a mathematical question. It's not a question that is going to save anybody's life. But I think there is a lot to appreciate around it, and especially the power of dynamic programming, but just the power of algorithms for being able to do something and compute something that at first may not even seem solvable. So that's what we're going to get. We're going to start much simpler than that today. We'll have a warm-up question. And I think the first three questions that we're going to do actually are pretty straightforward compared to what we've done already. So they're going to be baby steps compared to what we've already seen. The last three problems are going to be our challenging ones today. And fortunately, the idea of those problems is when we solve the first one, it's going to heavily inform the second one, which will heavily inform the third one. So if we just looked at the third question, it's going to seem very, very challenging and impossible. And the idea of these is starting with the hardest question instead of starting there in terms of solutions, building up to it. And we're going to see this idea of progressively going from there. Sarah, great to have you here. Jack, good to see you. Welcome from Upper St. Clair. Not too far away at all. Hope you're doing well. All right. So we're about to get started. As I said, this is going to be a full one today. A few reminders before we do. Starter code is always in our description if you want to follow along. There's links to that. We also have links to the code that I'm going to complete here, so you can always find that there as we go. This is also part of a larger initiative called Philomath. We run not only this particular collection of streams, but we have our Programming for Lovers course that we've been building out, and we're going to have more courses coming soon. So hang on to that thought for now. A lot of what we're doing here is going to be heavily related to the types of questions that we address in Great Ideas in Computational Biology, which is coming soon. So keep on the lookout for more information around that. We're finishing up our semester right now at CMU. So there's been a lot to do in the old day job, but we're fortunately at the end of the semester. So we're going to have a lot of time and energy for a lot more cool things coming to you over the summer. But for now, I would say, please, as always, make sure you like and subscribe. It keeps us visible. We've got more information in our description about joining our Discord, joining our mailing list, and you can always join Philomath as a member to go deeper if you're interested in an ebook. We have programming projects associated with programming for lovers and so forth. But in the meantime, as I said, this will be a full stream today, so I think it's fair that we should get started. As always, if you have any questions, let me know. A lot of times folks ask questions or want clarifications about code. I usually handle that after I go through the solution of everything, but let us know in the chat if you have any questions or just anything you want to say. Happy to have it. And I think we're ready, so let's code. PPER is the partial permutations problem. And we've worked with permutations up to this point in what we've done. We've talked about them as, for example, orderings of genes. So we have n genes on a chromosome, and the number of ways to order them is going to be n factorial. This question, though, is about ordering partial permutations. So in particular, say that we're only interested in ordering k of the n objects. And the example that we would give here in the problem description is, say we've got the first 8 positive integers, and we're interested in only a permutation of 3 of those things. So we need to pick the 3 things, and there are 8 choices for the first element, 1 through 8. Once we've picked the first element, there's 7 choices remaining for the second, and then once we pick the first 2, there's 6 choices remaining for the final one. So the statistic that we're interested in here is how is it we take that idea and generalize it to say, if I have an arbitrary value of n and an arbitrary value of k, what's the number of ways, in other words, what's being asked for in the return statement here, what's the number of ways to order k of those n objects? And we, as we've seen before, we have a modulo of a large number just because we're going to give you values of n and k that are going to produce a large value of this statistic pnk that we're talking about. And that might be a challenge with some programming languages to actually represent a number that big. But if we take the remainder when we divide by 1,000,000, as we go through and do the arithmetic, we're going to continue taking the remainder by 1,000,000. And that way we can know that you have the right answer. At least you'd have to be very lucky to get it to match when we divide by 1,000,000 and take the remainder without having to worry about large integers that we might pass into our solution. So this problem, I would hope, would be a quick one. We have PPER.py. And the idea is we have n total items. We have k items to choose and arrange. And then we have the modulus. So even though this is asking us for everything when you divide by 1,000,000, taking the remainder divided by 1,000,000, we're going to make that just a parameter to our partial permutations function. That way we could generalize this. And if we don't pass in, this is really nice, because if we don't pass in any input here, well, it means it's going to allow us to compute this statistic that we're interested in without actually performing the modulus. So without actually taking the remainder. We will see that shortly. I should say, for now, I might not even write the problem in that way. So I might save it here. In a future problem, I'll add it back. But for now, I'm not going to worry about the case where the user doesn't add a modulus as input. I'm just going to implement the function as we go. And the idea is that, well, there were n factorial total permutations, but in this particular case, we have n choices for the first element. We have n minus 1 choices for the second, and then so on and so forth. And we just need to make sure that we appreciate that we're going to get to the kth element. So we're only picking k. We're picking like 10 things out of 100, for example. And if we were picking 10 things out of 100, well, we would go 100, 99, 98, 97, 96. It's tempting to think that we're going to go down to 90 as the last element, but it's a classic like fence post counting question. So it's not n minus k choices for the kth element that we're going to order. It's going to be n minus k plus 1. So in the case of n being 100 and k being 10, you can see that by saying, well, this will be 100 choices and then 99 and 98. You can count that out. If you're going to have 10 elements, the last one you're going to consider has 91 choices, not 90. And all of those choices, once I've made this choice, all of these choices are independent of each other. It doesn't matter which thing that I pick. And once I've made all of these, it doesn't matter which element that I pick for the kth element either. So that's nice. Because of that independence, it means when we count these things, we're simply going to be able to multiply them together. So the answer is n times n minus 1 times n minus 2, all the way down to n minus k plus 1. This allows us to practice just, I think, some pretty introductory control flow. We set a value of result equal to 1. We're going to return the result at the end of this. And if we were solving this without a modulus, without performing this division and taking the remainder, we would say for I in range. And let's be fancy. Let's start arranging at n. Let's go down to n minus k plus 1. And let's decrease by a step size of minus 1 each time. Except that we need to remember that when we do this, this element would not be included, and we definitely want this value to be included in the product. So as a result, I'm going to take it one extra value so that we make sure that n minus k plus 1 is included. So in fact, we do go to n minus k, except n minus k will not be included as one of our values I. And every time I do that, I'm going to say I'm going to multiply the current result times whatever the current element I is. Of course, that doesn't have the modulus built in, right? So we could return the result and then divide by mod and take the remainder. That's what our percent operator is for. But what makes that challenging is, especially if we're thinking about a language neutral approach and something that we have seen already, is that when you do this, you're going to get a big number and then you're going to do the division and take the remainder, and you kind of defeated the purpose of trying to avoid generating a huge number because you've already done it, and then you just do this at the very end. So instead, what we're going to do is to perform these modulus operations, these percent operations, as we go. It turns out we'll get the same end result if every time I multiply the result by I still do this, but then I take the remainders every time through the loop as we go. And this is based off of some results in number theory, or I would just essentially say like elementary algebra, which is that if you multiply two numbers together and then take the remainder when you divide by something, you could do it separately as well. So you could take the remainder when you divide each of those numbers and then multiply them together. So you wind up with the same end result. And it means you don't have to multiply them together and then take the remainder. You can just, every time you perform multiplication, you keep iteratively taking the remainder as you go. So it's a little bit of a trick, but at the end of the day, you just get the same end result, which is good. And that's the problem. We can run this, and in this particular case, it's PPER.py, so I'm just going to run Python 3 PPER.py. And I get 51,200. That's what's expected. That's good. So I'll comment out all of the parsing code that we have in our stub. And then I'm going to download a data set. And I'll place that data set into my file explorer on VS Code, so it's in the same folder as what we're working with. And then I'll rename it, of course, dataset.txt, since that's what it's always looking for. And we commented out the parsing part. So when we run again, hit up and run again, the second answer we get here is going to go to output.txt. And that's going to be the answer on the data set that we're given. Notice it has to be an answer between 0 and 999,999. And so when we go back to the platform, everything looks good. We just need to copy in our answer, and hopefully we get it right. And we get a solution in record time. So I said this was going to be a lengthy stream, possibly. There's a lot to do with these problems. And you might be thinking, well, that was a quick one. That's probably one of the quicker answers we're going to get for any problem. So hold on to that thought. We're going to just continue peeling the onion back. But for now, that problem is good. We'll look at PMCH next. If you have questions, let me know. But PMCH will be our next question, so I'm going to get everything ready for it. Julia, hi from Belgium. Thanks for joining us. Good to see you. All right. Let's move on to the next one then. PMCH is perfect matchings and RNA secondary structures. So there's a motivation for this problem. A lot of times I think we don't get into the biological overview of these problems, but in this case, there is I think a simple biological concept, which is DNA can base pair. We know about that. The double helix means that A binds with T and G with C. But a natural question you might have when you learn about RNA, or a single-stranded version of DNA, is, can RNA base pair with itself? And the answer is yes. And this very frequently will actually occur when an RNA molecule is produced. G still wants to bind with C, and A with, well, the version of T that we have in RNA called uracil. And so this process will happen, and as a result, a lot of times you form what are called secondary structures. And if you have worked with proteins before, proteins have secondary structures too. So as proteins fold, a lot of times they fold into these recurrent shapes that then get combined into larger shapes when you get the final form of the protein. But these sort of intermediate shapes, which often are like sheets that run in parallel directions or helices that are formed as part of the protein, these are called secondary structures, in large part because we see them so often. And in the case of RNA, we get these types of structures too. In the RNA world, probably the most frequent one is something like this that's a hairpin loop. So you have the RNA is going in this direction, it loops around, and you don't have really much base pairing here, although you could have U against A and U against A. But then you see a lot of consecutive positions where you see a lot of base pairing. So there's a natural question here, which is to try and appreciate, what are the matchings for a given RNA string that could be produced? And folks who appreciate this as, this is a hallmark example of something in computational biology, because you have things, in this case, they're letters in our RNA string, and then you have connections between these things, which are this base pairing interaction. And anytime you have that, it makes sense to study this as a graph or a network. And so what we have here, even though it's a biological question, is inherently, like inseparably related to the concept of taking nodes in a network and pairing them off. And that's called matchings. So we talk about the concept, there's a lot of background here, but the platform talks us through this concept of a perfect matching, which is that if a network has an even number of nodes, the idea that we would be able to pair off the nodes, and then in some way we're going to have n total edges that pair off the nodes. Now, if we are allowed to pair any two nodes, then we can count the number of ways that we could pair them off. So if I pick my favorite node, well, it has 2n minus 1 options for what it can get paired against. And so you pick one of the 2n minus 1 remaining nodes. Then you have 2n minus 2, so pick your favorite one. And then it has 2n minus 3 possibilities for what it can pair off with. And you continue this process. You get what's called a double factorial. So the number, if you can match anybody to anybody else, then the number of matchings grows pretty big. It's 2n minus 1 times 2n minus 3 times 2n minus 5, and this continues falling until you can notice all these numbers are odd, until finally you get times 5 times 3 times 1. So it's the same type of counting that we saw in PPER, where once I make a choice, the remaining choices are free, and then you can multiply all these possibilities together. But in this particular case, the next question that you might ask is, well, what if it's not possible? Because here is the formula, if anybody can match with anybody else. But there are lots of possibilities when we want to match things. There are classic algorithms for matching. For example, you might have a dating site, or you might have a website that's pairing physicians with places that they might do a residency or something like this. And you want to associate, each possible node in one sense with a node on the other. So there's ways in which to generalize this. And the natural way of generalizing this is to say, well, what if it's not possible to connect certain pairs of nodes that are, like, for whatever reason, incompatible? And that's the case in the corresponding network for an RNA string. So when we look at an RNA string, we're going to still have a network. Our RNA string here is UAG, CG, UGA, UCAC, and so forth. Our network is the nodes of this string. And our edges are not going to be every node to every other node. Our edges are going to be represented by the possible base pairing relationships we could have. So we're going to connect a U to every occurrence of A, as you can see here, and even to its adjacent neighbor here. But we're not going to connect a U to a G or a U to a C. G's are going to be connected to C and vice versa, and those are the only pairs we're going to have. So in this particular case, you can see here's in the RNA example, this idea that some edges are restricted, some are allowed. And our first question, our question is to try and study this a little bit. What are the questions that we might ask about it? And our first one is to say, well, if you give me the RNA string, what are the number of matchings that are possible? So, in other words, what are the numbers of ways that I'm going to allow you to bond with A and connect C's to G's and allow all that to happen, except I'm going to specify that the matchings are going to need to be what are called perfect. So in this, what I'm calling this bonding graph of the RNA string, we want to count the number of ways to match C's to G's and U's to A's where everybody is getting a partner. So here is one particular way of doing it. And it turns out, I think for this example, you can see there's going to be a lot of other ways that we could have done this. So a simple way would be to say, well, I could do this G to C, and I could do this G to C instead. So we should expect for an RNA string, this number probably should grow because the longer the string, the more we're going to be able to match things. And we demand that this matching is perfect. Meaning, if I have, you're going to see we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 characters in our string. That's even for a good reason. And it also has the property that every A is going to have a corresponding U. Every A is going to have a corresponding U. Every A is going to have a corresponding U. So the number of A's should be equal to the numbers of U's, and the number of G's should be equal to the numbers of C's. If that's not the case, it's not going to be possible for everybody to partner with everybody else because we're demanding that this base pairing only occurs between pairs. So there is what we would call an invariant for this particular problem, which is the number of A's is going to be equal to the number of U's, and the number of G's is going to be equal to the number of C's. And when we demand those things, that's going to force the length of the string to be even, because you count up the number of A's plus the number of U's, you're going to be even. Number of G's plus the number of C's, you're going to be even too. And so the total number of characters in the string needs to be even for this to work. The question becomes how to solve this question. And our insight is that, well, this is related to finding the number of permutations up to some number n. Before we even get to that, I would say we have another insight too, which is the number of choices of A to U matchings is independent of the number of G to C matchings. And I think that's very reasonable, right? Because how I pair off the A to U's is completely separate from how I pair off the G's and C's. So changing an AU matching won't ever affect G to C because the allowable edges that I can use are completely distinct between AU pairs and GC pairs. So it's as if I actually have two separate networks here, or two separate problems. They seem like they're part of the same network, but actually they're kind of not, because the A's can only be connected to U's and G's can only be connected to C's. And you could imagine just taking your network and splitting it out into two separate networks and solving each of these questions separately. Whenever we have something like that and we're counting something, we know that the idea is going to be we're going to count the number of AU matchings, and then we're going to count the number of GC matchings, and we're going to multiply them together. So at the end of the day, anytime you're counting something, you can split it into two separate counting problems that are independent. At the end of the day, you take the number of things in one camp, the number of things in the other, and you multiply them together and you get a final answer. So then the point becomes, well, we simplified this down. How many AU matchings are there? And the next insight would be, if there are n A's and n U's, okay, well, let's imagine starting with one of the A's. It has to have a U partner. Okay, so the first A has n choices for U partner. The next A has n minus 1 choices. The next has n minus 2, and all the way down. And this time we're picking n things. So finally, the last A has only one choice. Okay, and the total number then is going to be n factorial. So we can use the fact that we can figure out exactly how many matchings there are going to be by counting up the number of A's. We might also verify it's equal to the number of U's. As I said, for this problem, we don't have to do that because the data that we're given, we know we're going to be in this particular form that have equal counts of AU and GC. So I'm not going to go and validate that. It would be a natural thing to do. But for now, what I am going to do is to say, well, what we should do is count the number of A's, count the number of U's, count the number of G's and C's. So we will assume that number of A is equal to the number of U and number of G is equal to the number of C. And that's going to mean that, well, let's just say that there's an A count which is equal to 0, and a G count, which is equal to 0. And I'm not going to count the other things. What I'm going to do is I'm going to range over the bases in the RNA string. If that current base is equal to A, then I'm going to add one to the A count. Else if the base is equal to G, I'm going to add one to the G count, and that's going to be helpful. Probably want to check that we have a valid RNA string. So it only consists of A, C's, G's, and U's, and that the number of A equal number of U and number of number of G equal to the number of C. But for now, I'm going to assume that that's okay. I'm more interested in the counting side of this. So if I'm interested in the counting side of this, what I'm going to say is I've got the number of A's, I've got the number of G's. Remember, if I have n A's, the number of ways to perform the AU matchings is just going to be n factorial if there are n total A's. So now n is equal to our A count. So AU matchings is simply equal to math.factorial of the A count, and the number of GC matchings is equal to math.factorial of the G count. And as a result, I need to import math, but the problem is now solved. And I don't even really need these variables themselves to represent the AU matchings and the GC matchings. I could just sub these down to here, but I think it's useful to think about the problem in this way, because if we don't write out these temporary variables, you might miss the idea, which is the critical idea, which is if I ever have two things involved in a counting process and they're completely independent of each other, the total is equal to the product of the two constituent total counts, which in this case relies on something that we have already seen, which came from the problem PERM. Once we understand that, it's just a factorial. We can get that factorial function, of course, from the math package. So we can save this and run. And when we do so, we're running PMCH.py. We get a value of 12. That's what's expected on our input RNA string. So far, so good. It means I can remove the comments around our parsing part of the code. I can go to Rosalind and download a data set. I can put that data set in my appropriate place. Let me make sure that I have the file explorer up here. Ooh, didn't mean to do that. We'll name it dataset.txt. And now I'll run my code again. And I get this big number. So in this particular case, and I think it might just be because of the scale of the data sets that we give, for this string, it's not a very long string, we have an enormous number of perfect matchings for that string. So let's make sure that this is right, but the idea is to demonstrate what might otherwise and would otherwise come up in a lot of disciplines, especially biology, but what is commonly called combinatorial explosion. The idea that something might seem like there aren't too many of something and that we can manage how many of something there is. And on this small input of length 10, we see that there's 12 possibilities. In this particular case, we did indeed get it right. And we see that there's an enormous growing number of possibilities, even for an RNA string that's not actually that long. Even though this is a counting question, and we're not getting our hands dirty with, studying RNA secondary structures themselves, that's really what this problem is attempting to communicate, which is that a lot of times when you are presented with a biological structure or something in practice, it might seem obvious or that it's going to be easy to figure out exactly at the end of the day how these things are going to base pair. But what this problem communicates is just enumerating all of those possibilities would probably not be the best idea because there are so many. And yet nature, at the end of the day, winds up picking one. It's this beautiful idea. We see this in the context of protein folding as well. The number of possible ways that a protein could fold is astronomical. And yet in all of your cells, in trillions of cells going on all the time, nature has evolved an algorithm that it just sort of produces a final folded protein with extremely good fidelity over and over again without any intelligence or any sort of underlying algorithmic drive. And the same thing happens with RNA secondary structures. So the RNA is produced and then it folds together into a final shape, even though there's this huge number of possible different folds. MMCH is what I'm going to do next. Let me know if you have any questions. Otherwise, I'm going to start here on this question momentarily. All right. MMCH is maximum matchings and RNA secondary structures. And what it is getting at is that, well, we've solved the problem of counting the number of perfect matchings in these RNA secondary structures. But one of the assumptions that we made when we solved PMCH is that the number of A's is always equal to the number of U's, and the number of G's is always equal to the number of C's. And that's a very strong assumption that in practice is not necessarily true. So you might assume that that's always going to be the case, but that's certainly not going to be the case, that if you have an RNA string that gets produced, it's got this perfect balance of the A's equal to the number of U's and the G's equal to the number of C's. So you can see in the sample, even before we explain the question, here's an example where there's one A, but there's 3 different U's, and there are 2 C's, but there's only 1 G. And so the question becomes how to take what we've done with perfect matchings and generalize it to this example of matchings that are as good as we can do. And if we're saying, well, it's as good as we can do, figure 1 here shows the left RNA string has a perfect matching, but if we change the U to a C, we've thrown off the balance of U's and A's and G's and C's. So it's not going to be possible in this network to form a perfect matching. But we can form what's called a maximum matching, which is if we look at this bonding network. The maximum matching contains as many edges as there possibly could be. And it's not, I don't think, too difficult to see and appreciate how many edges this is going to contain. We look at the number of A's in our string and the number of U's in our string, and we know, well, if there are 17 A's but there are only 2 U's, the best we can do is to have 2 edges. And if there are 3 A's but there's 14 U's, the best we can do is that we can only have 3 edges connecting A's to U's because you only have 3 A's. And there's a general formula, which is, well, the number of possible maximum matchings in this network is just the number of edges in a maximum matching is the minimum number of A's versus U's plus the minimum between the number of G's and the number of C's. So in this particular case, there's 1 more C than there are G's. So the number of edges connecting C to G is the number of G's that we see in our string, which in this case is just equal to 3, but there are 4 C's. Similarly, we have 1 additional A, so the number of edges connecting A's to U's is the number of U's, which is the minimum of the number of A's and the number of U's. However, notice we still have independence. So just as we had when we were looking at perfect matchings, we still have this idea that the number of ways to create a maximum matching, which is what this problem is ultimately asking for, is equal to the number of ways to maximally match A's against U's times the number of ways to match G's against C's. And so if we want to count all these matchings, what we're going to do is at the end of the day, we will still have that we're going to return the number of AU matchings times the number of GC matchings. Of course, we need to realize how is it that we're going to obtain a value for AU matchings and GC matchings. Well, we know that the number of edges in any maximum matching is represented by this formula, which we said is the minimum of the number of A's and the number of U's plus the minimum of the number of C's and the number of G's. We also know that when counting the number of matchings, we have independence between the AU matchings and the GC matchings. That's what allows us to go ahead and write this at the end of the function. Then it's just a matter of saying, well, in the case of AU matchings, if it was perfect, so there were n A's and n U's, we knew that the answer was n factorial. So when there were n A's and n U's, the number of AU matchings was n factorial. So maybe what we're going to have here is similar to that, and the answer to that is yes, which is good. But we need to write out maybe on an example what we're doing. So if we have n A's, or let's say 5 A's and 2 U's, then we get to say which of the 5 A's get connected to each of the 2 U's. So you pick one of 5 A's for the first U's partner, and then you pick one of 4 remaining A's for the second U's partner. And the answer there would be, well, 5 times 4, which happens to be equal to 20, but I'm more interested in how I get there. And in general, if you have like n A's and k U's, where I suppose we're assuming here that n is bigger than k, or bigger than or equal to k, then the number of choices is the same. You would pick n things for the first A that you're joining a U against, and then you have n minus 1 choices for the remaining one, and n minus 2, and so on. And it's tempting to say, oh, it's times all the way down to 2 times 1, but that's not right, because we only have k choices that we can make, because you only have k possible edges. And what we realize here is, well, we have the partial permutation problem in disguise, that at the end of the day I have n things, and I'm picking k of them, and I'm ordering them. So it looks different, yet at the end of the day, what I have is n A's, and as I go through the U's, each U's partner, each U gets to pick a partner. The first one picks n, the next one picks n minus 1, the next one picks n minus 2, so we get to use this independence again, except that we know what the formula here is. The last element is going to be n minus k plus 1, because we solved this in the PPER problem. Okay, so we get to reuse our solution. Of course, I want to, get to this solution, and the first thing that I need to do is first declare a dictionary to store the counts of nucleotides. So it's not like when we worked with PMCH, we could just say, yeah, count up the number of G's, count up the number of C's, or I think it was A's and then G's. We didn't need to pay attention to the others. But in this particular case, we need to know how many of each nucleotide there are, because they're going to differ, possibly. So I'm going to have a counts dictionary. It's going to map strings to integers, and we're going to start it off as not just, having A's are going to go to 0, C's are also going to go to 0, and then we're going to do the same thing for G, and we're going to do the same thing for U. And then what we want to do is range over the string and update the counts. So we'll say for base in RNA, counts of base is going to get plus equal by 1. So now our dictionary is complete. And once we've computed the dictionary, I'm actually going to declare some variables that are just going to make things a little easier to work with. So I'm going to say the number of A's is the counts of A, and I'm going to do the same thing for each of the other bases. And that way I can just say, you know, once I have done this, I've got these kind of intermediate variables, but what they mean is I no longer need to think about the dictionary, and I've got T, it's going to be U, but you get my point. I'm in DNA land. At this point in time, the dictionary is no longer needed because I've used it to produce what it is that I care about, which is the counts of each of these base. What we would like to do then is simply to import the partial permutations and say AU matchings is equal to, we've written this function in PPER that takes N and K and gives you this answer. So I don't need to rewrite that function. I'll say partial permutations of something, and that something is, well, what's our value of N? It's an AU matching, and when I did this analysis before and I said, well, the number of A's is bigger than the number of U's, you might have the opposite case. It might be that the number of U's is bigger than the number of A's, in which case you still have the same formula, but N is equal to the number of U's and K is equal to the number of A's. So it'll allow for that possibility. What we really want to say is that, you know, in each case, N is equal to the maximum occurrence of the two bases, and K is equal to the minimum. So I want N and K here, and I want to do the same thing for GC matchings of N and K, but N is going to be equal to the maximum of the number of A and the number of U, and K is equal to the minimum of the number of A and the number of U, and I'm going to do the exact same thing here, except I'm going to replace A and U with G and C. And now we've got a complete solution, except for one little thing, which is that when we look at PPER, it takes this required modulus, which is a little bit annoying, and there's a couple of ways of getting around this. So one thing I could do is I could give a number to this function as a modulus that's so big that it's unreasonable to expect that the number of permutations would ever go over it, meaning when we divide by that number and we take the remainder, we're just going to get the number back. That's, in my sense, a little bit kludgy, and it would be better instead to just make our function in PPER a little bit more robust. And in order to do that, I'm going to make the modulus optional. So I'm going to replace, I'm always going to take an N and a K, but I'm going to replace this third parameter by saying it could also just not be handed in. And I'm going to replace this with saying it's either an int or none, and it's also going to get a default value of none. So if someone doesn't specify a modulus, that's fine. That means that I won't use a modulus, and instead I'm just going to do the number of permutations of K objects from N. Everything will be good. Of course, that means that I need to update this here. And then in this loop, I will say, as I go through the loop, I'm going to say result equals result times I, or better, result times equal I, a little bit tighter there. And then I'm going to check. I'm going to say, I want to do this second step, but I only want to do it if the mod is not none. So if there is a modulus that I care about, I'm going to do this. In this way, we could go through, and I'll leave it to you to verify that this is not actually changing the solution that we had before. So we can still perform the modulus if we need to, but if it's not given as input, this is never going to get touched. So we're never actually going to be taking this remainder when you divide by mod, and you're just going to be multiplying by I each time through the loop and then returning the result. So it's a nice, tight, clever solution to this issue that is largely just a kind of a platform concern, which is that we want you to be able to generalize solutions to one question and then use that for other questions, and we've seen that quite a bit already. In this case, it's just a little bit annoying, and here's a way that we can get around it. Once I've done that, of course, now I didn't specify the modulus, and so we should be in good shape. At the end of the day, we return the number of AU matchings times the number of GC matchings, and we should then be able to run Python 3 MMCH.py. It should give us our expected value of 6 on this particular RNA string, which notice is not balanced between the numbers of A's and U's or G's and C's. That means that we can also have our parsing code go to the platform, and I think we should be comfortable with getting a data set and putting our data set over in our folder, calling it data set, just for consistency. Running again, and now we get this huge number because, as we saw, we're not enforcing the modulus for this problem. And if we had it to do all over again, maybe we should. That would allow us to have a little bit of consistency here. But I also think what we're getting at here is an appreciation for even the fact that this is not a balanced RNA string with number of A equal to number of U and number of G equal to number of C. And yet at the end of the day, we're still getting a huge number here. Assuming, of course, that number is correct, which it hopefully is. All right. So we're halfway through our problems for today. And we're going to have 3 more. CAT. So let me make sure that we can see this one. And then we're going to have MOTZ. That one's not going to be ready yet. And the third problem is going to depend on that one. So here's where we're going to turn up the difficulty just a little bit and solve our last 3 problems that are all going to fortunately build on each other. All right. If you have questions, let me know. Our next question is CAT ID. Rodney, welcome. Glad you could join us. CAT is up next. And as you can see, we've got a little bit of work to do here. And we've got to set up the problem. All right. CAT is Catalan numbers and RNA secondary structures. We've worked with RNA structures, secondary structures before, and we've connected them to this concept of matchings in a network where we arrange all of the symbols of the RNA string around a circle, and then we connect points of C's to G's and A's to U's. And in one problem, we looked at perfect matchings, where we have a string where the number of A's is always equal to the number of U's, and the number of G's is equal to the number of C's. And in those types of strings, when we match up A's to U's and G's to C's, we match them in this perfect fashion, meaning every node gets used in one edge. But it turns out that, biologically speaking, it's much more natural or more common for the base pairing interactions within an RNA secondary structure not to cross, which can be represented visually in terms of our base pairing graph here that we have by noticing that the edges themselves here never literally cross. So if we draw them as straight lines, then all of the edges that we have, and I could have drawn them as these straight lines, but all the edges that we have are not going to cross each other. That would produce, I think in some cases, in some sense, a tangled RNA secondary structure. And what we saw before with the hairpin loop, that's not the case. Okay, so with the hairpin loop, everybody matches with a partner without any sort of crossing of edges. And there are examples of RNA secondary structures that demonstrate that sometimes you actually do get crossings. So if you imagine you put 3 nodes or 4 nodes around a circle, and you connect 1 to 3 and 2 to 4, you're going to get a cross. And that happens sometimes in RNA structures with these structures called pseudoknots, but they're less common than what we've seen with hairpin loops and other types of RNA secondary structures. So if we're interested in studying the more common ones, well, what we might be interested in is to forbid any of these sort of possibilities where we have crossings. The greater challenge is to say, well, how do I know, like I can look at something and see that there's no crossings in an example like this, but how is it that I know, like how to define what that means precisely? And we can get at that if we think, well, imagine what would happen if you crossed over 2 edges like this. You would have edges that when you go through, you would have a collection of 4 points. And what is nice about these 4 points currently, and why we know they don't cross, is that I is the smallest index. If we're going clockwise here, I is the smallest index, and it's connected to, let's say, L here, which is the biggest index. And J and K are the middle indexes, and they're connected to each other. So if you ever had the case that I was connected to K and J was connected to L, that would be bad. So you also could have it where I is connected to J and K is connected to L. So if the smallest index is connected to the biggest or the second biggest for this collection of 4 indices, you're not going to have crossings. Whereas you will have crossings if you have 1 connected to 3 and 2 connected to 4. That's what we're really trying to get at with the picture of the pseudoknot, that you have sequences of RNA symbols, but they cluster together into this being 1, the next one you encounter is 2, the next you encounter is 3, and the next you encounter is 4. And notice you've connected 1 to 3 and 2 to 4. So those are the types of structures that we want to avoid, and we can be very fancy here by saying there shouldn't be edges connecting I to J and K to L where I is less than K and it's connected to J. So notice I said 1 cannot be connected to 3. So this would be our bad circumstance where I is less than K is less than J is less than L, but I is connected to J and K is connected to L, which is what you see here. So what's elegant there is that we've taken this visual concept of what it means for edges to cross in this network, and now we've represented it as a purely like algebraic, mathematical way of representing this statement. So that is a hallmark, I would say, in general, of quantitative science. It's why this type of field exists in the first place, because, and will work in other problems with things like evolutionary trees, but it turns out that in that particular case, there are beautiful examples of being able to demonstrate sometimes there is no tree for a collection of data, and we can write out in a very precise manner, algebraically, when it's the case that a collection of data are never going to produce a tree that fits it perfectly. That's, of course, the subject of another problem and another day. But for now, the idea, I think, is hopefully there, that we can take a concept that we think about of, an RNA secondary structure, and we can actually represent it purely algebraically in terms of the network that we're interested in. So we're extending the problem that we've seen before with looking at perfect matchings. So we're still going to assume that the string that we have is balanced in the sense of the number of A's is equal to the number of U's and number of G's is equal to the numbers of C's. That is good. But in this particular case, we're going to demand that there are no edges that cross. And that's going to complicate the problem quite a bit. The question is, are there still going to be a lot of these non-crossing perfect matchings? Or are we going to narrow it down quite a bit? In the case of our example in figure 3, there's actually only 2 non-crossing perfect matchings for this RNA string. Now, our idea is to connect this to a problem that is related, but that I think arises purely from a mathematical context. In the mathematical context, the easiest network to solve this on is one in which everybody is connected to everybody else. So if everybody is connected to everybody else, that means anybody can be a partner of anybody else. And the number of matchings that you can obtain that are non-crossing, if it were the case, for example, for the RNA string that A could base pair against G, or U could base pair against C, or U could base pair against another U. If we allowed all of those edges and then say what's the number of perfect matchings that don't cross, we would get something called the Catalan numbers. So the Catalan numbers say, if I have the number n, the integer n, and then I make this collection of 2n nodes, and I allow anybody to possibly be connected to anybody else, what's the number of perfect matchings of those 2n nodes that don't cross? And they appear not just here, but in a huge variety of other settings. So I'll say, if you're interested in them, go check them out. They arise in surprising places. And people who count things really love the Catalan numbers. They're often a favorite, because in all of these different contexts of how many of this thing are there, you wind up with the answer being Catalan numbers. Another, like, maybe non-biological example is the number of ways to valid, like the number of valid ways of parenthesizing with a lot of nested parentheses, parenthesizing a sentence or something like that. The Catalan numbers arise in this context, which seems like it's, disconnected from this, but they come up all over the place. And we're not going to compute the Catalan numbers here because there's a formula for them, but we are going to learn from how that formula could be computed. And it turns out that there is a closed formula, which is absolutely beautiful for the Catalan numbers, that is very hard to explain. It's a lot like when we looked at FibD, I think, where we had mortal Fibonacci rabbits, and we wound up with a formula like that. Actually, it was a recurrence relation in that particular case, but understanding the recurrence relation was really challenging. It didn't have a nice, natural, intuitive explanation. In the case of the Catalan numbers, there is a formula that says the nth Catalan number is equal to this formula. But understanding why that formula is true is very hard. Fortunately, there is a way of understanding the Catalan numbers via a recurrence relation that does make sense. So I want you to imagine that we pick our initial node and we want to connect that node to some other place. So if we're free to connect to any other place, I'm going to say connect it to point K. So you've arranged all of your 2n nodes around a circle, and the first one is going to get connected to node K. Then you've divided your network out into two pieces. I mean, we have a visual here. We're not allowing every possible node, but say C was our first node. If it's connected to this node over here, G, then you've divided out the number of non-crossing matchings that involve this edge as equal to the number of ways to have a non-crossing matching over here and the number of ways to have a non-crossing matching over here. And lo and behold, what are those two things? They're independent. And so you can multiply those two things together. So as a result, we say allow K to vary over all of the nodes. And when K varies, what you can say is, well, for a fixed value of K, once I know that, the number of non-crossing matchings is going to be equal to the number of non-crossing matchings over here, times the number of non-crossing matchings over here. And then we're going to take that product and we're going to sum over all the different values of K. So when K is this, I'm going to get a product. When K is this, I'm going to get a product, and so forth. Now you might imagine what happens if I connect these things, because if that were a valid edge, I only have 1/2. And if 1/2 is empty, I'm just going to say, okay, well, there's no possible, or there's one matching if I have no nodes in it at all. But the idea is that I obtain this recurrence relation, which is that C sub n, the nth Catalan number, is equal to, when you sum k equal 1 to n, you're going to say, well, node 0 is my first element. And so I have k minus 1 choices for 1/2 of this. So this is one side of your network where you can say you're computing the number of non-crossing matchings, perfect matchings of k minus 1 nodes, and that makes sense. So in this particular case, if this is node 0 and this is node k, you have k minus 1 nodes over here. And then you say, well, how many do you have over here? Well, it needs to be kind of like n minus k, and that's what you have on this side. So you multiply those two numbers together, but remember, k can vary, so you wind up with a sum for every possible k of this value. We would like to take that idea and generalize it to the idea of RNA strings, where I'm finding non-crossing perfect matchings, but I'm not allowing everybody to connect to anybody else. If I could join U's to U's and G's to A's, then I would just have a Catalan number problem, but I don't. I have this biological flavor of a Catalan problem that we kind of invented for this particular problem. So the question is how to count that. And our idea is, well, we are going to borrow the same idea. So we understand how to solve the Catalan recurrence. And with Catalan, I'll just reiterate, point 0 must connect to some point, say k. This splits our matching problem into two subproblems. So number 1, the number of non-crossing matchings from 1 to k minus 1. And number 2 is the number of non-crossing matchings from k plus 1 to 2n minus 1. We can multiply these together to get, and I'm going to say f of n, or f of 2n, is equal to the number of non-crossing matchings for n nodes. And when we multiply together these two things, well, this is the number of non-crossing matchings from 1 to k minus 1. So this is, 1 is f of k minus 1. And the second thing goes from k plus 1 to 2n minus 1. And this would be f of 2n minus 1 minus k plus 1. And then you would then add 1. Okay, so you'd say how many elements are there in this range? How many nodes are there from k plus 1 to 2n minus 1? So you just subtract them and add 1. And when you do this, you get a minus 1 here. This winds up being a minus 1 here and a plus 1 there. So those are going to go away, and I just wind up with minus k. Or I could rearrange it in this way to say 2n minus k minus 1. The point is, you get a product of this, and then the recurrence relation is f of 2n is equal to the sum of k from 1 to 2n minus 1 of this product that we have before. So I'll just copy it in. And that's very elegant and beautiful and clever and all that stuff. But the problem is we need one more thing, which is that there are base cases. When n equals 0, you're going to say f of 0 is equal to 1. This is an empty matching, and it's going to make sure that when I do this, if I ever get f of 0, I'm just going to say that's equal to 1, because I'm not interested in that side. I don't have any nodes on that side. And I don't want to zero out what this is. So this is a convention as much as anything else. And when n is equal to 2, you're going to say f of 2 is equal to 1. And that's easy. So that's, there's one matching if there are two nodes. And that sets up the Catalan numbers. So we could write this as our own function, and I would encourage you to go for it. The Catalan numbers have a known sequence. It would be good practice, and you could produce a function called Catalan of n and build it up in this way. But the problem is that when we extend this to RNA, there's an issue, which is that even we will assume in this case that the number of U is equal to the number of A and that the number of G is equal to the number of C. All well and good. However, when we extend to RNA, we also have the problem we can't have anyone connected to anyone else. So we're restricting what the matching can be. So one way that we could say that is if we look at the first symbol of the string, this is what our idea was before. Point 0 must be connected to some other point in the network. And in this particular case, if I said, oh, well, this means the first node, meaning the symbol associated with the first symbol of S, that has to get connected somewhere, right? And so it can only pair with K such that two things are true. Number one, well, S of 0 and S of K are complements. So that has to be a valid edge relationship. And then there's a second important part here, which is that, well, K minus 1, the number of remaining nodes, must be even. And the reason why is if I connect these two things and I split out and I've separated everything into two pieces, well, the things on each side must have an even number of nodes left. So the other way of saying that is K must be odd. And so our recurrence is going to be quite a bit more tricky. And our recurrence is going to be similar, but it's going to involve two things. In this particular case, I'm actually going to have two inputs. I'm going to call my function cat. My inputs are going to be I and j. And cat of I and j is going to be the number of non-crossing matchings, I should say, between nodes I and j. So if we just look at that isolated occurrence, and what we're really interested in then is we're interested at the very end of this process, we care about cat of 0 and length of RNA minus 1. But we're going to need the two inputs there because note that some of these values are not going to be valid, like e.g. cat of I to I plus 3, I plus 2, let's say, because we need to know that when we compute this, we need to say the number of remaining nodes must be even. When I do this, I'm getting a substring of length 3, which means I would be connecting I against I plus 2, so that should be 0, right? So there's going to be a lot of values that are just 0, and that are not allowable. And what we want to do is we want to make a table that's going to represent all of this, where at the end of the day we care about this, and our recurrence relation is going to be based off of cat of ij. And we need to be very careful with this, because when you do the Catalan numbers, you only have one variable as your input. It's great. It's easy because you can just compute the Catalan numbers as you go up. So when you're trying to set f of 2n, the idea with our recurrence relation is I've already computed these values. So it's straightforward. It's like computing a fancy Fibonacci. But in this case, I actually have two inputs that I have to care about, and now things are going to get tricky. And so our recurrence here is going to be, well, cat of ij is I'm summing over all k, but the k have to be within this range. So I'm interested only in the number of non-crossing matchings between nodes I and j. So I'm restricting myself to that range. That is the sum over all of k in the range I plus 1 to j. Okay, and I mean technically it's not including j, right? But we think about indexing in this way where we never include the top element there. But where k is odd, so that's important. Remember, that's something that we care about. And S of I and S of K are complements. Okay. And notice all of this is what we command. So we demand all of these things to be true. And then once we have done that, then we're going to have our function. And our function is still going to be the same, kind of, except we're saying I'm interested in everybody from I plus 1 to k minus 1. So I'm connecting I to k by virtue of doing this. But if I do that, then I have the number of non-crossing matchings from I plus 1 to k minus 1. And then I multiply that times the number of matchings from k plus 1 to j. Okay. So I have represented the problem as kind of a sub-problem, right? I'm solving the number of matchings between these two positions in the string, and I still actually have basically the same end result recurrence, but I just make this demand that I'm only looking at values of k where you have k as odd, but also S of I and S of k can actually base pair with each other. We also need to say what are the base cases. So what are the base cases here? And the base cases here are a little different because you want to specify, well, if I have the same input in each case, cat of I and I should be equal to 1. So this is like connecting to itself. It's an empty matching. So there's no nodes in there. And we just are going to set that equal to 1 because when we do this product, we don't want to multiply something that is a genuine value by a value of 0. And once I've set that base case, I'm also going to say cat of ij should be equal to 0 in these weird cases, remember? So I said if it's the case that j minus 1 is odd, and you're looking for the number of non-crossing matchings between these two positions in the string, so for this substring from I to j, if j minus 1 is odd, that would be bad. And this is going to include j. So the idea here is, if you go from cat of 0 to 4, you're talking about a substring of length 5 there, because I'm going to include the j for this example of the recurrence. So we have 5 elements in our string. That would be bad because j is 4, j minus 1 is 3, which is odd, and that's the number of remaining symbols after a matching. So now this becomes completely invalid. So it's a handful, frankly. And to solve the problem, I think we just need to learn from this Catalan recurrence and be careful with what we're doing here. But we're going to learn from something that we've already done as part of this series, which is we have a dynamic programming recurrence. And to compute that recurrence, we're going to build a huge table. And funnily enough, we're getting a two-dimensional table, just like what we had when we produced longest common subsequence recurrence, when we worked with the edit distance. And so we're still going to get a two-dimensional dynamic programming table, but what it represents is the number of non-crossing perfect matchings for this substring, going from position I to position j. Okay. So as we write this, it's just a matter of taking this idea and converting it all down. So I should copy this down, actually. And I'm just going to put it beneath here so we can look at it if we need to. But we start with needing to know the length of the string because I need to know that how big is my table. And I'm in the wrong place. I need to have this next to the table. That would be a good thing to do. Okay. So it's going to be n plus 1 by n plus 1. And we will say, all right, n is equal to the length of RNA. That's probably a good thing to do. And we're going to make a table. It's going to be blank. It's going to be a list of integers because it's counting things. It's going to be empty. And then I want a range over everything up to and not including n plus 1. So this will give me n plus 1 values. I'm going to make a row, which is a list of integers. And it needs to have a bunch of 0 values. As we said, a lot of these values are going to be 0, so they're just going to never get set. And then I'm going to append to the table that row. So the first thing we want to do is make our table. And the next thing we want to do is handle our base case. And so to handle this base case, what we'll say is, if I is equal to j, it's 1, and if j minus 1 is odd, it's 0, but odds are we're just never going to set this value. Okay. So let's handle this case, and I'm actually going to do it in this way. I'm going to say, let's say if it's an empty substring, also have a value of 1. So for I in range over n plus 1, this is ranging over all the rows. And then empty substrings, by this I mean what would happen if I is bigger than j. The idea is when I'm computing this CAT of ij, the assumption here is that j is at least equal to I. So if I is bigger than j, that would be bad. How do I access those values? Well, I would say for j in range over I, so this is if j is smaller than I, that would be bad. And I'm going to set table of ij in all of those cases to be equal to 1. All right. So far, so good. Then it's just a matter of saying, well, we're going to fill in our table in order of increasing substring length. And I think I've not actually said, so the 0 values are handled here. I believe it's going to be okay just to go up to I plus 1 here. So that's going to set table of I equal to 1 as well. So let's do it that way. Then we're going to go through and fill it in order of increasing substring length. And the reason why is that when you look at our recurrence, okay, at the end of the day, when this thing gets computed, what we really care about is that these two values have to be computed. This is something that we saw when we worked with dynamic programming before in the context of sequence alignment. I can't set a value with a recurrence relation unless anything that recurrence relation needs to access, like predecessor values for smaller inputs, have also been set. And you think, now we're working with this recurrence relation, I need to make 100% sure that when I try to access cat of ij, I've already set all of the values in this formula. And in order for me to make sure that that's true, it means for any k, both of these have to be set. What is nice that I know is the case is that, well, these are smaller substrings. Why? Because when I look at cat of ij, this is going up to k minus 1 from I plus 1. So this is k is somewhere between I and j. And k is somewhere between I and j here too. So both of these are going to be smaller, meaning the top index minus the bottom index, or the length of the string that I'm working with here, is going to be smaller than the difference between j and I. So that's why this is going to get filled in order of increasing substring length, so that when the recurrence relation is called, we know that values have been set. Okay. So we will build our way upward. And so to do that, I'm going to say for a length equal to in the range of 2 to n plus 1. So we're going to start off with the allowable range of values being equal to 2, and then we're going to go up by 2 each time. That's going to prevent us from considering possibly looking at computing the number of non-crossing matchings in a substring where we have an odd number of things. That wouldn't make any sense at all, right? So if we have an odd number of things, there's not going to be a perfect non-crossing matching in that range because somebody's going to get left out. And that's why I'm going to range my lengths of starting at 2 and moving upward. I start this range equal to 2 because we're going to set small values. So we start length equal to 2 because this is going to indicate like j minus I will start small. I don't have j minus 1. J minus I will start small. And when I set that length equal to 2, then it'll go to 4 and 6 and so on. I know what I is, so actually what I want to do is I'm going to start I in the range of everything up to n minus length plus 1. Okay, remember n is the length of the string. And I do this because I don't want to set a value of cat of ij that's like well outside where j is too big. Okay, and the reason why is I need to set j. J is a function of I by saying it's equal to I plus length minus 1. So this ensures that the substring from I to j has even length and the appropriate length, or length equal to length. Yuck. Okay, so that's the length of our substring. And when I set j equal to I plus length minus 1, that will ensure that that's the case because I'm looking for the collection of values between I up to and including j. Okay, normally with substring indexing we would do it this way, but cat of ij includes j, so we're going to say minus 1 here. All right, and then once we've done that, we're going to store a variable called total, which is going to be eventually what we set, because down here I'm going to set table of ij. That's our cat of ij is equal to total. And then way down here, then I'm going to return the table. So I'm filling in an entire dynamic programming table for this, which I will return down here. But then here's where I will apply the recurrence relation. And the recurrence relation says, look at all these possible values of k. And what are the possible values of k? Well, it can't be I. Okay, so it's going to start at I plus 1. And then it needs to go up to, its allowable values. It could be j. So j is an allowable value for me to base pair against, if it's valid, right? If I'm base pairing an a against a t or whatever. But the other thing that's important is that, well, I know that I can pair up with what do I have? 1 plus 1. That's 2, I think. I know I can connect I to I plus 1. That's its neighbor. But I can't connect it to I plus 2, right? Because then I would be leaving out one nucleotide that's like, well, I can't connect to anybody. I would have to cross, right? So if I connect I to I plus 2, I'm leaving one node in the middle that would have to cross that edge. So as a result, you can go to I plus 3. So I can go I to I plus 1 or I to I plus 3 because then there are 2 elements there in the middle. As long as that's allowable, that's all right. So I'm going to jump up by 2 each time. So this is, remember that we said k in some sense, we need to ensure that it's like even from the perspective of I. So, it's going up by 2 each time. And then once we do that, I'm going to say, is k a valid partner? Because if not, I don't want to consider it at all. So if it's possible that they actually pair, and what do I mean by they? I mean, I'm taking I, and I'm trying to connect it to node k. If I can do that, then I want to say, well, okay, access the 2 elements of the recurrence relation. And when I access those 2 recurrence relation elements, what is it that I'm accessing? I'm accessing this thing times this thing. So it's the function from I plus 1 up to k minus 1, and then the function past k up to j. So that's good. I'm just saying I know how to get those. It's everything on the left side. This is 1/2 of the data, which is the number of crossings between I plus 1, or matchings that don't cross, from I plus 1 to k minus 1. And then the other half of the data that I care about is access the table starting at k plus 1 and going up to j. And remember, independence. So the total is equal to its current value plus the left times the right. So left and right get multiplied together, and then I'm going to add whatever that is into the total. Now, there is a little bit of an annoyance here, which is I also have a mod on this question. So we haven't had it for previous matching problems, but the mod has made an appearance again. And so because that's the case, I need to say total equals total plus this product. And that's an unnecessary parentheses because of order of operations, but that's all right. At the end of the day, I want to percent by mod every time I go through. Okay. And now I set the table value equal to that total after I've ranged over all possible k's. Here is where the recurrence relation actually happens, by looking left, looking right, and then doing this. Of course, then I take the remainder after I divide by mod. And I'm kind of done because there are two more functions here. But as we've seen before with dynamic programming in the context of sequence alignment, once I make the table, then I just need to look at the value that I care about in that particular table. So when I make the table, so I'm going to have two things that I need to write. I'm counting up the non-crossing perfect matchings. And the first thing I want to do is make the table. And that's going to have the RNA and the mod that I care about. So I just pass those two values into the function for non-crossing table. And then I want to return the bottom right element, so to speak. So even though it's not a sequence alignment problem, we're doing the same thing, except it's not even the bottom right element. I guess in some sense it's like the top right element, because what I'm looking for is the table value corresponding to the whole string. So it's the element of the table corresponding to the whole string. And how I've done that is, remember that the first element is the starting position, and the last element is the final position. And so the starting position is 0. The last element in a lot of cases would just be whatever the index of that table is. But in this particular case, it's I want to return the table starting at 0, ending with the last position of the string, which is length of RNA minus 1. And then we're in OK shape. So this function actually only has 2 lines. And then there's just one more thing to do, which is I have one more little subroutine called can pair that takes 2 bases, and that we called it here, somewhere in here, which says I want to make sure that k is a valid partner. When I'm ranging through and I'm jumping up by 2 positions every time between I and j, I'm going to say, is this current position, is it possible for it to pair with the ith symbol of RNA? And if it is, then I perform the recurrence. If not, I'm just ignoring it. It's not going to contribute to the total because that's not a valid edge. So I need a function that takes 2 bases and that simply returns true if those bases are valid pairs and false otherwise. So this should be a quick function. What I will do is I'm going to say, you know what, let's just join the 2 pairs together. So this is a tumor, not a tumor like a cancerous tumor, but a tumor in the context of bio, meaning a substring or a string of length 2. So we're going to concatenate base 1 and base 2. And then I want to return true if that pair is like AU, UA, CG, or GC. So I've got a string, and I can do this with just one line by saying I've got a tuple AU, UA, CG, and GC. And if that pair of concatenated symbols is in this tuple, I want to return true. If it's not, I want to return false, and that's the one-line way of handling it. So that's our function. Now, do I assume that this is right? Maybe. I don't know how confident I am in my ability to live code this, but let's see. We have a data set here where we have this string AU. We should expect 2. And when I run python3 cad.py, we get 2. So that looks pretty good so far. I'm going to comment out what we have, and that could be a complete fluke. So we'll download the data set, and we'll copy it over. And we'll call it data set. Rosalind cat is a nice data set name. Should have kept it that way. And we get 239680. And let's see. Okay. Whew. Right? That's not an easy problem to explain, I would say. And it's because I basically tried to teach you the Catalan numbers in like 4 minutes. And those are, I think, a reasonably advanced concept in math. I would say as a math undergraduate student, I think I saw them as a junior for the first time. And then we worked with them even in a PhD in math. But this is part of the inherent cruelty. Biology is the cruelest discipline. Certainly among science, but probably in general. And it only makes sense for us to have a Catalan problem that's actually way harder than the Catalan numbers themselves. I don't know what the moral is of that. The fortunate thing is the next problems we're going to see are also super hard, but they're not like the jump is not going to be as big, right? So we're going to take what we've done and then we're going to go from there. And you'll see it's going to be very similar. If you have questions, let me know. Next problem is going to be MOTZ. All right, 2 more problems to go. MOTZ is Motzkin numbers and RNA secondary structures. We've seen Catalan numbers, and Catalan numbers are if you have 2 n things, meaning an even number of things, and anybody can pair with anybody else. What's the number of ways to do that, such that if you arrange them around a circle or in a line, but you arrange them around a circle, it cannot be the case that there are any crossings of the edges that you see. So when you match pairs off, they can't cross. And we saw that in the case of Catalan numbers, where if you allow anybody to pair against anybody else, you have a nice recurrence relation. And we extended this to the concept of RNA strings, where we're looking at RNA secondary structures, and we're going to allow ourselves to have base pairs, but we're going to demand that C has to, pair with G and U has to pair with A. However, we have seen this example in the context of perfect matchings. So we want to generalize this a little bit where we're going to allow ourselves to have non-crossing matchings. We're still going to demand that the matchings do not cross. But we're going to demand that these are all possible non-crossing matchings and not just perfect ones. So if we look at a particular example here, in this case, this will allow us, I guess, a little bit to not necessarily assume that the number of U's and A's are equal and the number of G's and C's are equal, but we're leaving out possible connections. So G doesn't necessarily base pair with C here, and U doesn't base pair with A. So we have a matching. It doesn't necessarily use all the edges it could, but it is non-crossing. And we introduced this problem because we solved this for perfect matchings previously. We saw it had a connection to Catalan numbers. And this is kind of just a little extra add-on to say, if you don't demand that the matchings are perfect, and then you want to count up how many there are, well, there's going to be more because you're getting all the perfect matchings, but you're also going to get matchings that are not perfect. So if there are 5 labeled nodes, for example, and this allows the input to be odd, not even, because you're no longer demanding that everybody has to have a partner. You're actually going to get 21 different ways of forming a non-crossing matching. So the number here could be enormous, right, and grow very quickly. In our case, if we were solving this particular problem, saying anybody can connect to anybody else, there's a recurrence relation here too. So the first node that we connect, it might not be involved. Before, when we had the Catalan recurrence, we said the first node, it has to connect to somebody else. So pick a partner for it. But it may be the case that it doesn't actually pair with anybody. So it might just be on its own. And if it's on its own, you've essentially taken that node and you've flung it into the cosmos. And what's left is n-1 nodes. So then you're saying, well, how many matchings are there of the remaining n-1 nodes? And if you're representing the formula that you care about as m of n, m being named for Motzkin, whoever that is, then you know that on the one hand, you could have m of n-1. And on the other hand, you could have used this node that you picked in some matching. But if you use it in a matching, then you're going to get something that looks a whole heck of a lot like the Catalan recurrence that we had before. So if it's used in a matching, well, you have to pick a partner for it. And when you pick a partner, they're going to connect, which means you divided the problem into 2 halves, which means that you have, the left half and the right half, and the number of ways of matching the left half and the right half are independent, so you're going to get a product, but then you sum that over all possible partners. So this was the Catalan recurrence. The only thing we're adding here is the possibility of saying this particular node might actually not get used. So the problem seems way harder in the sense that we're not demanding that the matchings are perfect. They could just be anybody, and it might be the case that nodes don't get used, but it turns out at the end of the day, the recurrence relation winds up being extremely similar, which means that computing the Motzkin numbers is very straightforward. If you want to compute m of n, you just need a one-dimensional table where you've already computed all these values, and then you access m of n-1, and then you access all of these things and multiply them together, and then you set m of n. So computing a Motzkin number is not hard. As with what we saw with the Catalan numbers, with just a couple minutes of explanation, I can tell you how to do it. Go do it if you like. What we're going to do is the thing that, because it's biology, is of course way harder, which is to say, let's just connect this problem to a case where not everybody can pair with everybody else. And in the RNA string world, when we have our base pairing graph, that's going to be the case. So when we look here, right, it's legal for C to base pair with G, but it's not legal for C to base pair with C or U or A. So you have to have CG pairs and you have to have AU pairs. And that's going to mean that we need to be careful here because we need to specify that that's the case. There is a fortunate thing that's not the case in this particular case, which is, imagine that you had an A here. Okay, so this were an A. Before, when we looked at the version of this problem for perfect matchings, we had to be very careful because we had to say, you cannot base pair with this position because you would be leaving yourself with only one symbol left over, and that would be bad. You'd have an odd number of things over here. Similarly, you couldn't go from here to here because you'd have 3 elements over here. And that's mean because you have a perfect matching. It's not possible to have a perfect matching with 3 nodes in it. 2 of these things can pair off, and then 3's a crowd, as they say. So if that's the case, it won't work for perfect matchings, but I want you to imagine for matchings that are just general, certainly this is okay. You could go to G, and A just gets left out. A is just not going to have a partner. Or you can go to G, and maybe all 3 get left out, or maybe 2 of them are going to pair off, and 1 gets left without a partner. Either way, this is going to be valid. So this problem seems trickier because we're counting a larger number of things, but at the end of the day, actually this aspect of it where I had to ensure that like K was even compared to I is not actually going to be important, which is good. So I'll borrow my idea of my 2 cases from this Motzkin recurrence that we have here, and I'm just going to generalize it to the context of the RNA string when we're going to make our table. So I will say, we should write out what we have here, which is, we borrow the idea of the 2 cases from Motzkin. And I'm going to write my function as MOTZ of ij. This is still the number of non-crossing matchings in the bonding graph of S between positions I and j inclusively. And once I say what that formula is, then the hard part is to write it out as a recurrence relation. So we're going to say MOTZ of ij is equal to, all right, I might not pair with anybody, all right? I don't mean I, or maybe I do, but I am or I is going to be lonely. And in either of those cases, what I mean is I is or am getting left out of the matching. So I'm interested in saying this is the case where I'm flung out and I'm not considered part of the matching. But then I also have the other case, which is the sum over valid K of, this expression, MOTZ of I plus 1, K minus 1, times, so this is, need a Z there, so this is everybody from I plus 1 up to K minus 1, so this is the left half, and then the right half is everybody from K plus 1 up to J. So this is assuming that I've connected S of I to S of K. And if I have the left half and I have the right half. Great. Now, the valid K here is a lot easier. So the note is that we no longer need that even odd check for K, vomit. But we do need for S of I and S of K to be complements. And you're going to have base cases. So if I is greater than or equal to J, then MOTZ of ij is equal to 1. So that's what we saw before. So either this is ridiculous because you're looking at a substring that doesn't exist, I to j where j is before I. So we're going to set it equal to 1 in that case. We're never going to be accessing those values. Or I and j could be equal, in which case you have the substring, the number of matchings of me to myself, and I don't match with anybody, but that's in a sense a matching on its own. So I'm not connected to anyone, but I'm also just going to say, well, for the sake of this recurrence relation working out and to be nice to myself and have a little self-love, I will say that the number of matchings involving me is 1, even though no one's actually getting connected. So that's an easy case to handle. When I go to the non-crossing matching table, I need to say the same thing that we did for cat, n is equal to the length of RNA, and the table is going to be a big list, and I'm being very precise with this because it's not hard to do so, okay? But it's ultimately just an empty list, but it's a 2-dimensional list of integers. And for I in range over n plus 1, this is all code that we had before. Row is a list of integers, and it's equal to a bunch of zeros times n plus 1. And of course, there are ways in Python to write this all in one line that are hideous and vomit-inducing, and I'll leave those to you too. But we will say table.append of row, and now we've got a bunch of empty 0 values. And now let's set our base case. So we'll say for I in range over n plus 1. So this makes it have the appropriate length. We're going to be n plus 1 by n plus 1, which is what we want. So now we're ranging over all the rows. And you know, if it's the case that j is less than or equal to I, set the table to 1. So I can do that with the same code that I had before. J ranges over everything up to I plus 1, and I set table of I, j equal to 1. And then we're good. So the table has been set. All that's the exact same. And then we're going to fill in increasing order based off of the length of the substring. So the exact same. Now we apply the same idea as in cat of increasing the length of the substring and computing a table of ij for increasing values of j minus I plus 1. Okay, so remember, table of ij is going to represent the number of matchings between positions I and j inclusively. Then it's just a matter of remembering this thing. Okay, so I'm just going to have that down here to help me. Okay, and I will say for length and range of 2. I can't type anymore. Range of 2 to n plus 1. Before we had comma 2 to make sure that we know that the length had to be, we're going up by 2 each time, but we're not specifying that that's the case anymore. So it is possible for us to consider a substring from I to j that has odd length. That's all right. But we're going to range from 0 up to the n minus length plus 1, because j is going to get set to be equal to I plus length minus 1. Okay. So this is going to ensure that j is within the bounds of the string. That's important. And once I've done that, then I'm going to say the total is equal to, all right, in this particular case, I'm going to set it equal to the table of I plus 1 j. And what I'm really doing when I say that is the total is equal to 0. It's just like it was the case in CAT. So when we're looking at the Catalan version of this problem. And then I'm going to add table of I plus 1 and j. So this is where I is lonely, meaning I am not including I as part of the matching. All right. Once I've set that, then, so I've handled this part of the recurrence by adding that into the total. And now I just apply our version of the Catalan recurrence from before, meaning all of this stuff, and that gets added into the table. So then we'll have the I is not lonely case. And if I is not lonely, then we're going to say 4k in range from I plus 1 to j plus 1. This is the same thing that we had before. So we're going to range at possible partners from I plus 1 up to and including j. And when we do that, what are we going to say? All right, well, if it's valid, so if it's a valid connection between RNA of I and RNA of k, and you'll notice I have imported can pair from the cat problem because I'm just going to reuse it. So this is just saying that's a valid connection. And if that is a valid connection, then I'll say, what are the two sides? I need to look left and look right, meaning I need this expression here, and I need this expression. Those are just table values. So I'm going to say left is equal to the table of I plus 1, Talby, the table of I plus 1, and k minus 1. And right is going to be the table of k plus 1 and j. And what I want to do is say total, not spotlight search, total plus equals left times right, But here's this little annoying part of the problem. And we could generalize mod to also have a value of none, because what I want to do is to say, I want to percent this by mod. So total equals total plus this thing and percent by mod. And that's good. The only other thing that there is remaining to do is to say, after I've ranged over all the possible partners, I've done both cases. I've done the I is lonely and I've done the I is not lonely case. And those two things are both added into the total. So we added them in here and we added them in here. And now that I'm added them both in, I'm going to set table of ij, in this case, equal to that total. And then way down here outside of everything, I've used my recurrence and I'm going to return the table. So as we have seen, the hard part of dynamic programming is knowing that it's useful, like that it's appropriate for this problem. Once you have identified that, the next problem is to write out your recurrence relation, which in this case is this, except k needs to be valid. But we're adopting this recurrence relation from a purely mathematical question to apply it to this biological context. Once I have the recurrence relation, you're like 90% finished. Yes, you have to get your ducks in the proverbial row in the table and make sure that you know what you're doing here, but you know it's a two-dimensional table from this, because we said, what does MOTZ of ij represent? It's the number of non-crossing matchings in this bonding graph between positions I and j. And it's represented by this recurrence relation. So then once I establish the base cases, I just set those, and then it's just a matter of going through and setting all the table values. Once I've got the table, then the value of the table that I actually care about is the same value that I care about before. So if I'm interested in counting the number of non-crossing matchings of this RNA string with this given modulus, all I need to do is, I even could copy this over from cat, but we have the non-crossing matching table. We give it the RNA string and the modulus that we are going to divide by and take the remainder. And then I want to return the table value corresponding to the full string. So the full string starts at position 0 and it goes up to and including position length of RNA minus 1, and then we're done. So all of a sudden the problem kind of snaps shut, and I can run Python 3 mods.py and get a value of 7. That's what we expected. I can therefore get rid of this comment here, grab a data set from the Rosalind website, and then put it back as dataset.txt, rerun our program, and now we get a value of 852556 for this data set, which we put back in the platform. And we get a correct solution. So that problem is a little funny to me, because even though we added this term, the problem is actually a little bit simpler, because by allowing myself to just sit on the sidelines, it means I no longer need to worry about the fact that I can only join to a valid partner if both sides have an even number of remaining nodes. That makes this Catalan problem a little bit more frustrating than MOTZ. It leaves our final boss for today, which is going to be RNAS. And if you're following along with this, you can see we should be proud of ourselves for being able to do this. You know, we're starting to see how many people are solving this. 5,400, 4,100, 1947, 1711, 1056, and 711. And who knows what will happen. Maybe all these numbers will jump up when bots take over the internet and they're like, oh, some of these problems are kind of cool. Or maybe they get bored and they only solve the first 3, but we'll see. But the idea is there's a big drop off here as we go through these problems. And this is definitely the problem, I think, that we have encountered RNAS that has the fewest number of solutions to it. So we're going to start hitting the rarefied air of the really challenging questions on Rosalind. And our next is RNAS, as I said. It turns out, if we can do everything else that we've done today, it turns out more than 700 people should have gotten this. That's often how these problems go. Okay. So RNAS is our last problem. Put in a breakpoint for no reason. RNAS is Wobble bonding and RNA secondary structures. And it's a little similar to the MOTZ problem where we were looking at Motzkin numbers and all possible matchings that are non-crossing in a network. And that network is a network of where for an RNA string, every symbol in that string is a node, and allowable possible matching edges connect A to U and G to C. This problem is probably the one that we would want to start with as an example of how dynamic programming can appear in contexts in biology that are not just sequence alignment, where they're ubiquitous. We would want to say we want to allow matching of edges. We want them to be non-crossing. We want to consider all possible matchings, so they don't necessarily have to be perfect or anything. The reason why is it may not be the case that A bonds with U and or the number of A's and U's is equal and the number of G's and C's is equal. But this problem is going to add two more little things to what we've worked with before. Number one, it's going to say we've been allowing very nearby RNA nucleotides to base pair. And that doesn't make a lot of sense. So the whole idea of allowing or studying RNA secondary structures is that it would be weird for an RNA secondary structure to very quickly tie back on itself and have the case that for two very, very close nucleotides, they're going to form this base pair interaction. If we look at a structure like a real RNA structure. So this is this bonding graph that we're calling it of an RNA string. It's a real non-crossing matching. It doesn't look like much on the left, but when we rearrange this and we infer from this what this thing will look like in a finely folded RNA structure, now this starts to look kind of neat and biological, right? So we start here at CGU over here, and we have CGG over here. So as we're going around the string this way, we're going around this RNA string in this direction. But as we go, we have a lot of these base pairing edge relationships that over here, again, are just connections across the circle. But when we redraw it in this way, we see that this looks like an RNA secondary structure. And what you see is something that's very natural, which is, for example, I'm going in this direction. This G is connected to this C. It's not connected to this C. It's not connected to anything up here, because if you're going to make one of these hairpin loops, you need enough space to actually make the loop and make the turn, okay? So as a result, you can't connect G to something so close to itself. It's not going to be able to loop back on itself so tightly. And so we're going to represent that by the specification that K, the connection, so if we're connecting SJ and SK, K has to be a little bit downstream of J. So we're going to demand that the partner of J has to be at least J plus 4. Okay, so that all that does is prevent these nearby nucleotides from base pairing. Then you have another thing, which is that in this particular case, we're actually allowing, so before we had A's and U's and G's against C's, but you'll notice we also allow U against G. So this is called Wobble base pairing, and we want to allow this as well. So even though the classic base pairs are U against A and G against C, sometimes in RNA secondary structures, you will see this Wobble base pairing of U against G as well. So we're generalizing what we've seen before. We're going to take an RNA string as input. We're going to return the number of valid matchings in this network for the RNA string. But we're going to assume also that Wobble base pairing is allowed, and we're going to demand that you can't be connected to anybody unless k is at least equal to j plus 4. This is the problem you would probably present to somebody as, here's a cool biological example of this. Everything that we've done with other RNA secondary structure problems has just kind of built up to this. And I don't think it really would be very fair to give someone this problem without giving them the context that we've already seen with maximum matchings and how to count things with independence, what the Catalan numbers are, what the Motzkin numbers are, and so forth. We've kept taking these small steps to build up to this. If presented with this problem to say, count the number of matchings here, I think a lot of people would just say that seems either extremely difficult or possibly impossible. Whereas the answer is anything but the case, that it's impossible. And it probably seems much different from what we've done because we have this extra condition of k is at least equal to j plus 4, and we're also going to allow for this Wobble base pairing that we see, but it turns out it's not. So I'm going to show you by going to our MOTZ problem where we did the same thing except we allowed for nearby base pairs and we also did not allow Wobble bonding. What I'm going to do is take all of my code for this non-crossing matching table. I'm going to do something very dangerous, which is to copy it over. I'm going to put all of this into what we call a Wobble matching table. And then I'm going to walk through it. N is equal to the length of RNA. All of this code is declaring our table. Our base cases are not going to change. So if j is less than or equal to I want to set the table value to equal to 1. The recurrence relation itself is also not really going to change, right? So the recurrence relation that we had before is represented by this Motzkin recurrence, where I say there's two possibilities. Either I get no partner as part of the matching, or I do. Well, all of that is still the same. The only thing that changes is this part, the summing over the valid k. In order for k to be valid, it can't be right next to me, so I have to go at least 4 units downstream, 4 indices downstream. And I'm now going to generalize the concept of base pairing to say, well, G could base pair with a U. So I want to allow for that too. And the point is, everything else here is the same. So the length ranging will be the same. I is going to be the same. J is going to be the same. And so forth. I actually would say probably, if we're being precise, we could probably increase this value, because if I'm looking for, the number of matchings between two positions and they're only separated by 2, well, there's no way they'll have any valid matchings because there's no way that I can connect 2 edges together because of this extra condition that they have to be at least 4 elements apart. And yet, let's just see what happens, because I think all these values are just going to be 1, meaning it's never going to actually set values, because if we handle this appropriately, then we may just consider the value of k is going to be outside of any kind of range. And so this is what I mean here, which is when we start the k in range here, so this line is still the same, but everything is related to, is k valid? And for k to be valid, you need 2 things to be the case. The can pair function here needs to generalize to say, well, I'm going to change this to can pair Wobble. And then when we range k, before we're ranging over all possible values, including the value that's right next to me. So I'm no longer going to allow that. I'm going to start my ranging at I plus 4. And that's going to ensure that if I have a partner, I is not lonely. I can connect to that partner, but only if it's at least 4 elements down from me, and then we can wobble, baby, wobble, baby, wobble, baby, wobble. But here is all the same. When we do connect, we have left is equal to the table value between I plus 1 and k minus 1. Right is the table value from k plus 1 to j. And everything else is set. That value gets added into the total. The total is set, table ij is equal to the total, and then we return the table. So it's just a matter of saying, well, yeah, I need to count the number of wobble matchings. And mea culpa, we don't have a modulus in this problem, so even that winds up being a little bit easier. We didn't have to consider this, I guess. And then when we count the number of wobble matchings, we will say, okay, everything else here is the same. We can even copy it over because I want to say the table is this wobble table that we have. So we need to call it the wobble table. We don't have wobble matching table. We don't have a mod. It's just RNA. And then we return this element, which is the number of valid non-crossing matchings between the first and the final position of my string. And we're good to go. There's only one other thing, which is that I have not written this function, and that function is going to be so close to what we wrote in cat.py. And in cat.py, I have my function that will allow, it'll say, can they pair? Well, I have 2 RNA symbols, concatenate them together, and see if that concatenated string is one of these 4 elements. I'm going to do the same thing, except I'm going to allow 2 more. I'm going to allow GU, and I'm going to allow UG. And now we're going to see what happens when I run Python 3 of RNAS.py. And we expect this value. We have a little bit of a cruel sample here. But hey, no worries because it matches. So because it matches, I'm going to try this, get rid of that. So we have our parsing code. Then we're going to download a data set, put it into the appropriate place. Oh, it already exists, so no. I need to make sure I remove it. And then we're going to run. And we get this number. And it's huge, especially because we don't have a modulus. And let's see. And just like that, with only changing 2 lines of code, we can take what we did before and generalize it to these 2 additional specifications that we have of preventing nearby nucleotides from base pairing and adding our wobble base pairing. That's probably it for today. I think there are some higher level learning objectives maybe that we've achieved. Number 1, I think the most important thing is that I got to use the word wobble more in about 30 minutes than I have used it in probably about 18 years. So that's always a good thing. And that alone makes me happy. But the other thing that makes me happy is that the problems are getting a lot harder. And we have plenty more to do with dynamic programming. We're not done there. Now that we're getting comfortable with it, we're going to come back to more advanced questions in sequence alignment soon. And we're going to come to more advanced questions in counting things soon. So we saw some pretty advanced counting concepts today. I hope they made sense. And I hope you appreciated that we can take something that's in a pure mathematical context and apply it to a biological one. And actually, the biological problem becomes, I think, just as interesting, but a little trickier. And that was nice. So we're going to keep the march ongoing. Things will get more challenging, but they're going to be doable. Next time, we're going to see that we need to learn a little bit more about networks. It's going to prepare us for more advanced studies, and we'll come back to more dynamic programming, but we've seen lots of dynamic programming recently, so we're going to look at more network studies next time. These run every Thursday at 11 a.m., so we'll look forward to seeing you next time for that. But that's enough for now. I can't wait to see you again, and until then.